taken off the market because saccharin was shown to cause cancer in laboratory rats, mice, I can't remember which. And that was controversial among the producers of saccharin because the experiments that were done there were feeding the animals a dose of saccharin for which you would have to drink 100 cans of soda a day, every day, for years to be able to achieve that level of saccharin. And we talk about why you do an experiment that way versus, so what you're doing in that case is you're sacrificing external validity, but in order to create a bigger effect size so that you can have a smaller sample size, which makes the experiment logistically feasible. If you're trying to detect cancer that occurs at a rate of 1 in a million, you're not going to have 10 million mice in your experiment. First of all, that raises the questions of animal welfare. It also raises logistical questions. I mean, even if you had a facility that could hold 10 million mice at $3 a day for bed and breakfast for a mouse, you're not going to be able to afford to do that experiment. So these things lead, one thing leads to another, leads to another. We lead these kinds of long discussions, and it's not, the way I do it is not formatted, right? I don't just read bullets down like I'm doing to you. I don't really do it that way. It's a discussion, and sometimes the discussion goes in some directions more than in others, but eventually we get around to all of these ideas and we develop them. So I'm going to describe, I've varied this project a bit from year to year. I'm going to describe what we did this last fall, this last autumn's class. I asked the students to test the hypothesis that eco-friendly products are really eco-friendly. Okay? And I'm showing you an example of some eco-friendly, here's a couple of eco-friendly, allegedly eco-friendly laundry detergents versus Tide in that example. A couple of things, if you're thinking about doing something like this on your, with your own students, a couple of tips. One, pick common household items for health and safety reasons. You can get just as good an effect from doing this with dishwashing soap as you can with using mercury. This is a lot easier to do. I have done it, we've done it, Nick and I have done this with de-icing salt with the students. There's a product on the market called Paw Safe or something like that. It's meant to be better for your pets, for houses that have pets. They test that against traditional de-icing salt. You can use laundry detergent, window cleaner is a good one, dish soap. I had a group use toilet cleaner before. I don't actually tell them what to use. I say it's got to be something you can buy at a grocery store. They can pick the eco-friendly products that they like to test. My constraints in this example that we used this year was they were allowed to have 60 pots. We used, I don't know, about that big pots. What would that be? 3-inch pots. And they have to use bean plants because that's what we gave them, bean seeds. We actually gave them a couple of other choices within that. We gave them, I think it was 3 or 4 varieties of beans that they could choose from, and 3 or 4 kinds of potting mixture. Those are really just red herrings for them to think about the extent of inference. If you do everything, you do all your work on one variety of bean in one soil, what does that say about the generality of your results? It's just something for them to think about. It's also something for them to trade off, because this is all about trading off generality and inference for replication and statistical power. And they get a lot of that as a design. Okay, so I really don't give them any more than that. I say you can have up to 60 60 pots, and you got to use some beans. That's what you got to do. Now the rest is up to you. Test that hypothesis. So they have to think about how many products are they going to test. Not just, am I going to do toilet cleaner and window cleaner and laundry detergent? Pretty much everyone quickly figures out they can't do those things. So they focus on something. I'm going to do laundry detergent. How many products of laundry detergent are you going to do? Are you going to do 1 eco-friendly and 1 conventional? Or are you going to do 3 of each? You're going to do 5 of each? What's the trade-off in there? What are you giving up and what are you gaining by making those decisions? Like I said, how many bean varieties? What different kinds of potting mixture? How many beans per pot's a good one? baskets for a discussion there about why would you ever want to have more than one? What strength are you going to apply? Are you going to just pour the laundry detergent straight out of the bottle and onto the soil? Are you going to apply it on the leaves? Are you going to apply it to the soil? Are you going to dilute it? If you're going to dilute it, what concentration? That usually engenders a lot of discussion about, as I say, about external validity. So, what concentration of laundry detergent would plants in the environment be exposed to? Which you quickly realize almost none. But even if it was, it wouldn't be the one that was straight out of the bottle. And there's some real experimental trade-offs in choosing to do 10 different concentrations versus you choosing to do 3 in terms of how many replicas you can have. And what do you gain by having that many different concentrations? When are they going to start? Are you going to plant the seeds initially and then immediately start putting laundry detergent on it? Or are you going to let them germinate first? Are you going to let them grow to a certain stage first? What's the pros and cons of doing those things? And how does that change the hypothesis you're testing? This is usually a good one, actually, in this discussion. What is the control in this case? We always have a long discussion because some groups want to use a concentration of 0, or in other words, they want to use just plain water as one of the groups, and other groups don't want to do that. What do you gain, what do you lose by including a water treatment, given that you have a fixed number of replicants that you can use? What are you going to measure? That's what I was going to say. Okay, you finally figured out how you're going to do the experiment. What is it you're going to measure? What's the impact going to be? You usually have to tell them, they want to initially count how many live. So you have to tell them, well, look, I'm pretty sure if you can buy this stuff in the grocery store and you can keep it in your house, it's probably not going to kill all these plants. So maybe you need to think of some sublethal measurements. But that's always good because it leads to a discussion about, you can say you're going to do this measurement, but how are you going to do this measurement? They'll say things like, well, we're going to look at how green the leaves are. Okay, well, how are you going to measure green? And how am I going to know, because that's going to involve some experience and some judgment, how am I going to know that when Kim measures the leaves, that she's getting the same answer as when Jonathan measures the leaves, and that leads us to a discussion of inter-observer reliability, and how do you control for that in an experiment, and et cetera. You can see that this conversation can take quite a while. I do this, so remember I've already kind of prepped the field with these couple of 2 or 3 lectures of experimental design and principles and ideas, very general. I'm applying this, I've given them, the moment they walk in, they have a 2-hour lab discussion class once a week. They come in, I split them into their groups, and I give them, here's the problem, here's your constraints, you guys have 30 minutes to discuss it in your groups, and then we come back and we have the critique. They'll present, and I'll say, okay, what have you thought about this? And while they're doing their discussion, I walk around, I eavesdrop, and say, yeah, you might want to think about that, or they'll ask me questions. And some groups are really inventive. It often has to do with whatever their home life has to do. One group managed to they brought in the material safety data sheets for the products that they wanted to use. That was very good. So we do the design and the critique all in those 2 hours. And usually they have not settled all of their ideas by the end. But I tell them, by next week you need to, I'm not discussing it with you anymore, by next week you need to have come to a decision on these things. And the next week they come. This year we did it in the greenhouse. We brought them to the potting shed, they potted up all their plants, and they did it on benches in the greenhouse. The last year when we did road salt, they did it in their dorm rooms. So if you don't have access to greenhouses, which I imagine many of you don't, this is an experiment that can be perfectly